I had a title, which is The Power of Viruses, but this is the subtitle. And I just wanted to show you that actually this can be our working environment. And it is kind of funny that I was supposed to talk 2 years ago, before COVID, and then this was really, you know, like, hey, what is a virus? What is it doing? And now you guys are like, yeah, virus, what is it doing? Had enough of those viruses, but I still like to try and engulf, take you with me on a journey to show the power of viruses, also the good power. So hopefully at the end of this evening, you share the same enthusiasm as I do. So let's first reiterate biological virus, just for those who missed COVID. We're talking about bundles of genetic material in a capsid. You can see in the right upper corner there, it's DNA, RNA in a nice hexagonal shape, sometimes with a landing, here with a tail and a landing platform, like here. And these landing platforms, they search for the right position on the host. But you can also have the engulfed viruses, the one lower, that actually just takes a little piece of the cell membrane of the host with it. They're very small. They're 20 nanometers, so that's 20,000, well, it's very small. But they can also be big, as I will show you in a slide further. Very big, by the way, so stay tuned. Their shapes are very interesting. Here we have the 3 domains of life, and that's not really so relevant for now, but I mean, come on, look at this. This is what we know. And then we have the potato, and we have, I don't know what it is, looks like a worm. We have some lemon shapes, some funny heads. And we're actually still discovering those. So there's plenty of interesting shapes and forms of them. They're obligate parasites. They're not alive. They are a very successful life form, but they can't reproduce on their own. So they're definitely parasites. They need a host to reproduce. So once we know this, let's go to some virus discoveries. It's really, seriously, not because I just want to pump you up. No, it's really a very interesting area to be studying viruses. Because we have multiple different funny discoveries, but we also have recently the giant viruses. And they're huge. You can see here, this is 400 nanometers, but that one is 1 micrometer. And just to think of, the bacterial, small bacteria in the ocean, they're around 0.3 micrometers, right? So these are huge. They have a tremendous amount of genetic material in them, way more than some of the cellular organisms that actually do reproduce by themselves have. What do they do with all these genes? Why is this, right? I mean, and the names get funnier by the minute. Because in virus taxonomy, there is no end. But you can't name it after yourself. That's forbidden. So the really interesting thing about these giant viruses is that they contain genes that until recently were thought to be only in cellular organisms. Right? So at some point, they have collected, or were they maybe live and they lost? That's a really debate that is ongoing at the moment. And we're finding more of these giant viruses. Personally, we have an algal virus, which is very small, giant, because it's only a third of this Mimi virus, but it has that same type of genes and the same characteristics. So at the moment, this is not my doing, but it's here, it says, for those in the back, it says that it's really an interesting time where the discussion is about what is actually life, what's a virus, is a cell with all these virus particles in it, can you still call that a cell, or is that actually, you know, maybe it's an organism. So we have to kind of like look at this at a very different angle. Just to kind of give you an impression how it looks from the inside, this is an algal cell. This is my little giant. It's called Phaeocystis globosa. You can find it in the North Sea. It's all over the place, especially in spring when you have the foam on the beach. That's the one. And what you do is you take a thin section, you cut through the cell, and you kind of like have a really high magnification, and you can see some chloroplast. This is a photosynthetic organism, so it actually needs the sun to make organic material, and you can see the nucleus, and you have a flagella base here. The other 3 are just cells that are infected. And very interestingly enough, this is 8 hours after infection, that's 12 hours after infection. You can see that there are some particles formed here around this kind of like sphere, this kind of like whitish center. And when you look at 12 hours, there's a whole bunch of them already. These particles, these hexagonals, are filled with DNA that stains very dark with these thin sections. And what we call this VF is a virion factory. This is where this particular cell makes its viruses. And it's not doing that in a way, like the nucleus is gone. It still needs the chloroplast, it needs some energy, but you don't really see a nucleus. So let's just extrapolate a little bit. We're alien life, we're coming to Earth, and we're finding this. Would you now think that this is the organism with the DNA of the virus? Or would you think, no, there's other DNA, I just don't know where it is, but it's definitely somewhere. So that is kind of like really, for me, it's kind of really interesting to see how these viruses can take over the metabolism of their host. And also in a good way, by the way. The other thing, and that's why it's important to actually show you this variant factory, the other super cool thing is that we now have viruses that infect viruses. Yeah? And it doesn't end there. We have now elements that infect the viruses that infect viruses. So this is getting crazier by the minute. This is the giant virus, and here are its little viruses. They're called virophages, and they parasitize, I can't really pronounce that word, this giant CoV virus, and actually this one infects an amoeba. It's one of these, those kind of like little unicellular animals, organisms, whatever it is, that makes all these shapes and kind of like any shape you want. So this is cool, right? And we have one in culture. And now this is hot off the press, literally, it says here somewhere down there, preprint. We're still under the review with this one. We found another one. Completely different, not linked to the virophages, but has that same kind of behavior and can also infect our giant PGV, our giant Viocystis virus. So we're finding things every minute of the day, and it's, as I said, it's a super exciting moment. However, there's always a however, this talk I'd like to talk you a little bit more to my core business, which is the virus ecology. And what is ecology? Ecology is in a way the organism with its environment. And if you talk about virus ecology, it's the virus with its host and its environment. And all these connections that it makes and the impact it has. This is, by the way, colored, not because it looks like that, because the journalists like to have color. This is the way it is. So we have these virus-host interactions under different conditions. We do that on cellular level, we do that on genetic level, but we also take it to the population level. We look at the biodiversity that is a resultant of these virus infections, because if you kill one, there's space for another one, right? We look at how the food web structure is changed. Is it changed? Also the energy and matter transfer, how is that actually affected? And then the other thing is really looking at these element cycling, the carbon, the nitrogen, the phosphorus. How is that going? How big is this? And that brings us to, why do we care about marine viruses, right? Because we're used to see that. We're very self-centered. We like to see the Earth as a lot of land and preferably a little bit more turned so that the Netherlands is a bit better. But if you turn it another way around, you see a lot of water and hardly any land. It's just the way you look at Earth, right? So planet water is better. Most of you probably know that 71% of the Earth is water, and of which 98% is seawater. You might not all know that the average depth is 3.7 kilometers. If you're very quick in math, then you can do the math, but this gives you a lot of water, right? 1.35 billion cubic meters of seawater. It's a huge volume. And we know there's a lot of life in the oceans. And with that, there is a lot of viruses. Global ocean, we have 10 to the power 31 virus particles. So that is 31 times 10 times 10 times 10, 31 times. That's a staggering number that nobody really has a perception of what it means. So hence, the 200 million we upgraded from the 100 million in the movie. The 200 million viruses per glass of seawater, and if it's an American glass, it's more. But this is also depending a little bit on where you are. In the coastal regions, it's higher than in the open ocean. Luckily for us and for everybody as a human being is that the viruses are very highly specific for their host. So if the sewage system of the country is fine, you can swim in the ocean without any problem, right? These viruses will not infect us. But let's just stay like with this glass of water, just a drop of water. How does that look? If we give this DNA, RNA, this genetic material, if we give that a green fluorescent color, then these are our bacteria, and all these little dots are the viruses. This is a little drop of seawater. This is not concentrated. This is just what you see under the microscope when you stain it. And it's just mesmerizing in a way, right? It looks almost like a starry sky. And of course, we have these really kind of like back of the envelope colleagues always, right? They kind of like scribble and calculate things. One of my colleagues actually did that, and he came up with this really nice comparison. If we put all the viruses, the 10 to the power 31 viruses, back to back, like a pearl string, we end up with 10 billion light years. Yeah? Just for comparison, I didn't know at the time, but the Milky Way has only 100,000 light years diameter. So if you then look what that means, these 10 billion, we go farther than the 60 galaxies. And luckily for us, the numbers are huge, but the biomass is really small. Otherwise, we would probably be drowning in viruses. But come on, this is just amazing, isn't it? I mean, just that pure fact, like, what are they doing? I want to know. Well, they're doing something. They infect every second 10 to the power 23, which is still a ridiculously high number. These infections take place in the global oceans, right? And that again is for journalists in colors, and that's actually, I forgot to give the credit to the colleague, I think it was Stephen Short. But it's kind of like, it is a bit like this, right? When you see the light on the ocean, it's kind of like these flickerings. That speed is approximately the infections that take place, right? I mean, it's just immense. Who do they infect? I mean, we know seals, they get the flu. Anybody who's having a fish farm or an aquarium knows that they can get sick. So yeah, they infect more or less everybody, right? And everything in the ocean as well, like on land. And not one virus, but there are multiple viruses infecting the same species. As you can see here, it's a really nice image with some of these little sea fleas, krill. There's a whale. What else? Crab. It's all kind of visible. But that's not the ocean. I call it the microbial ocean, because I think this is a real... If you take away this from the whole evening, I'm happy. 70% of the living biomass in the oceans, so you can see it, is microbial, right? So it's made out of unicellular organisms. 70%. So it's a bit like the iceberg. What we see above sea is a small tip of the iceberg. Majority is under the ocean, under the water. And here, literally, we can't see it. Literally, this is actually all these microorganisms that we need a microscope for. If you're really fast and smart, you can see that these are coins and not algae, which I originally thought. But I left it because it actually is really interesting. These microbes are our gold. They are absolutely essential for whatever we do. They form the base of the food web. It says under there, for you can't read it, I will say it anyway. They form the base of the food web. They're in high numbers, which is handy if you're a virus, because you cannot swim to your host. You need to wait until you bump into it by accident. So the more hosts, the more you bump into it. That's COVID, we all know about that. They have fast generation times, typically, except when you're in a deep ocean. And therefore, they also have rapid co-evolution. They are essential. Microorganisms are the drivers of any element flux, any cycling of carbon, nitrogen, sulfur, phosphorus, you name it. It's done by microbes. And therefore, they have a global impact. So for me, they are the perfect study organisms, but this is really why I do it. That's why I don't look at the seals, plus I will probably not win from the Dutch virologists, but this is really for me why I do this, right? So looking at the base of the food web, this is kind of like the typical standard food chain. Every step you go up, there is less biomass because they respire and they poop. Here you have your unicellular algae. We call them phytoplankton. So if you call me, say phytoplankton, that's my kind of like, work habit, but I now explain it to you, unicellular algae. They have hardly any biomass, 0.02% of the biomass on Earth, in contrast to land plants. But if you're in the ocean, it's really not very useful to have a tree trunk of 3.7 kilometers, right? I mean, the light is over here, this is the ocean, you need to be somewhere here. And that doesn't work, it's no use. you need to make sure that you stay in this upper water column. You can't get the light, so you need to be not too heavy, because then you sink out. So you reduce your size, unless you're in the coastal area, because there's some mixing, so you get up all the time. So you make yourself very small. The other thing why you make yourself small is that the nitrogen and the phosphorus that these algae need, like plants, it comes from the rivers, right? It comes from land. It's used by the algae in the coastal zone. So first we have dilution, and then they also used it all. So the ones that are actually in the open ocean are desperate, right? There's very limited conditions for them. So if you're very small, and your intake goes via your surface, so your volume is relatively small compared to your surface, that's optimal. You get a lot in, but you don't need that much, right? So you're tiny. That's why they're all unicellular, for these algae at least. Well, a food chain you cannot just endlessly go up in higher trophic levels. You need to kind of like bring it all back. That's by the degradation of the poop and the dying organisms, right? So this is the traditional food web, right? Algae, schematic, critter, and a fish. So this is what we have. This is what we're used to. The algae end up to higher trophic levels, as we call it. And this is what happens now if you have a virus infecting a single cell. It really doesn't matter whether you have the flu or whether you have a lethal disease. They're all lethal. You will die. And when you die, you release these viruses and the cell content, and you end up in little pieces. But the cool thing for us as ecologists is that this happens in this upper water column. So you've got all these mini corpses floating around, and they're degraded right away. And they bring back nutrients, nitrogen and ammonia, for the remaining algal cells. And they're all desperate, so they, thank you, right? Don't kill me, but kill the neighbor. They're nothing different than humans. So if you summarize this is what we call the viral shunt. It's a really nice word because it's shunting away, right? Viral shunts away nutrients and energy away from higher trophic levels. Doesn't go to here. This person actually had an original tropical fish, but a lot of my work is in colder areas, hence the Arctic cod, or I stand corrected if there is a fish expert in the room. But it doesn't go to the higher trophic levels. It actually ends up with dead cells and dissolved organic matter that is released, together with these new viruses that they produce. And that is, again, decomposed by the bacteria, brings back nitrogen and phosphorus into the system. So far, so good. Sounds cool. You agree? Yeah? But is it real? Do we measure this? Is this of any importance when you go in the oceans? It can be theoretically a really cool exercise, but we don't know whether it's really happening, right? So one thing here is that we have still very limited measurements. It's quite hard to measure this, to measure a mortality rate due to lytic viruses. Especially for phytoplankton, because most of the viruses actually kill the even more numerical dominant bacterial hosts. And then you are with your little phytoplankton group, right? Essential. But we do need to find original methods, which we found. And I'm going to show you 3 examples in this talk. So we try to do it all over the place, but it needs money. So we tend to have our research cruises are very concentrated, right? We try to do as much as we can so we can bring all these samples back home and analyze them. I'll go with you to the northeastern Atlantic Ocean, northwest Atlantic Ocean. This is in summer, here we go in spring. And we actually go full productive season to Antarctica. So let's go to sea. Now, this is our Dutch vessel. Somehow the image looks even more. It's a 65-meter ship. It's a super ship. It is very pragmatic, very Dutch. There is no space left. Everything is used. But it is actually really nice. There are no empty corridors. There's no, whatever. There's no pretending. It's just basic research. If you look good, I mean, it's a bit compressed. It's normally a little bit longer. But if you look good here, you see a container. Can you see that? These are lab containers. So when the goods are out, you put the people in. And this is how it looks. We pack up all our stuff in our lab, My lab has about 4 times this because we go on different places in the ocean. And then we still have the lab that we have at home also. So I've had a lot of pumps and I have a lot of aluminum boxes. So you pack it all up, you ship it to wherever the ship is, and then you unpack it and you make it into a lab container. Right? So you strap everything. You see everything is strapped except here. Clearly it was a nice weather day. Otherwise all these 1 liter bottles would have been on the floor. We make long hours. We work at the temperature of the water that we sample from. So here, not too bad. Sometimes when you're in Antarctica, it's very cold. Of course, we can't do the minus 1.5, so we're trying to work at 0. It's warm. So we do this with good weather. Yeah, this is our ship. We do this also with bad weather. Southern Ocean is quite notorious. Can have really nice weather, can have also really bad weather. We don't have icebreakers in the Netherlands. So we go with international collaborators. And seriously, I was on the bridge when I took this photo, and the wave came over the bridge, and I did ask whoever was in charge, so how high is the bridge? And he says, 17 meters. Okay, cool. This is really, I mean, the good thing with an icebreaker, it doesn't, let's put it this way. The waves in the Southern Ocean are not like the North Sea. The waves are like this, but they're high, right? But you can kind of ride them. So and an icebreaker rolls nicely, instead of, in a way, this ship would be kind of like really more like, if it would roll like an icebreaker, it would tip over, because it's made like that. Icebreakers are made like that. It's very different. But we were rolling once, and we were all like, are we ever getting up? And even the crew thought it was a very far roll at the time. So we sample. In here, there are sensors like temperature and salinity and an indicator for algal biomass and some other sensors. We lower it. On the way down, we registrate everything. And then on the way up, based on the profile that we see, we determine where these bottles, they're open when you go down, where we close them. Electronic signal, and then they close. So we end up with all different depths when it comes on board. And then we sample gently, and depending on what we do, for example, we look at genomics, or we look at metabolomics, or we look at whatever. In this case, we're trying to prepare for viral lysis assays where we need virus-free water. Really well strapped, as you can see. So this is kind of like preparing, and then once the assay runs, somebody is there measuring 18 hours a day. But we do shift. So let's go to the first example. We have this latitudinal gradient, northeast Atlantic. We go from south to north, and from 0 to 200 meters depth. Yeah? What you can see here is density. So this is by temperature and salinity that kind of like determines the density of the water. And the density here shows that it's not homogeneous. I hope you agree with me, right? It's not one color. It's quite layered. This is warmer water on colder water, and that stabilizes the water column. And that's called vertical stratification. And that process prevents deeper mixing going up, because there is hardly any mixing. And you can see that's very severe here in the south, and a little bit less severe in the north. If we look now at the nitrogen and phosphate, it's the same similar image. And this says 0, by the way, so everybody, purple, pink, whatever this color is, 0, right? Very, very, very low. So everything that is there has been utilized. Everything runs on recycling in this area. Here, a little bit less, still limited. On that where you see the larger algae, they can actually, use that phosphorus there. But here, in the south, you see that the algae, very small. It's very strange when you say very small, it's always like high pitch. Very small. Larger. But you see that they are not in the surface, right? That's kind of weird. But it's so limited here that they are trying to go down, trying to get that little bit of phosphorus and nitrogen that is over here. If they go down too deep, there is no light. Damn, they're photosynthesizers. So they need to find a place, and this is amazing in a way. This is 80 meters deep to 40 meters deep. They have hardly any light there. Open ocean, very limited. You can more or less see light down to 100 meters. And it's really scary if you ever swim in it. I could tell you. You have the feeling that there is always something under there coming up to you. I don't know. It's kind of. Some people are very calm with it, and I always have something like that. Where's this shark? Yeah, I'm a marine biologist, but this really fast open ocean is kind of weird. But we're not allowed to swim hardly ever so. But anyways, back to the story. You have these algae that are trying desperately to get a little bit of light and a little bit more nutrients. So we've got a nice gradient here. I hope you agree with me. Why do I care? A, I want to see if all these different algal groups, and that's here. This is an indicator for chlorophyll, a pigment that the algae have. And here's a size indication, and you can see all these different groups. So for each of these groups, we can determine the viral lysis and the grazing, so the predation on them. But I also study this transect because I want to know if there is viral lysis, is the share viral lysis grazing going to change? Why this transect? Well, the predictions by the ocean climate models are for the northeastern Atlantic that this vertical stratification will go faster, earlier in the year and more strengthened, more what? Pronounced. Thank you, more pronounced. Thank you, that's a good word for it. And that means that if you look at this picture, this is the way it is now, right? Stratified, but it's going to be more stratified. So we have less mixing, less input from nutrients from below, smaller algae. That's kind of like what the predictions are for our region. Well, I asked the funding agency whether I could actually stay for the ship for 50 years in the same place. Of course, I faked that phone call because I know what they will say, no. So you need to be a little bit more original, right? You need to think, okay, this is already happening. 2100 is already happening in the south. So if I look whether there is a change in south and north, maybe I can use the south predictions to actually say this is what we will see in a couple of years, because it goes faster than it was there. This is just to kind of like show you that we can do this for every group of phytoplankton. We can show where they are, very, very small ones. And some, you know, like everybody stays a certain place. What I really want to kind of like focus on my screen, it looks really bright red, but this is kind of red. Bars, that's the viral lysis rate. And the blue bars is the grazing rate. Now, if I ask you, and of course this is the latitude, again, from south to north. If I ask you, do you agree with me that there's quite a bit of red in there, or brownish, or whatever this color is here? Would you agree? Yeah, right, huh? And most of the stations have it. I mean, sometimes there is hardly any, sometimes it's dominating. But it's not like there is only a certain region or only a certain group. It really is kind of like everywhere. And that's a no-brainer once you know. But you need to show that first, right? The other thing is really clear from this is that on average, they're kind of equal. They're not really too different. There are more groups that we don't show here, but if you add up all the groups together, that's the result. You have as much viral lysis as that you have grazing. But this was the traditional concept. There is no viral lysis. We also measure gross growth rate. So this is what is possible without loss. Right? This is the potential. This is your gross income. Look, and then you take away the taxes and then there's nothing left, right? So this is the potential. This is what they grow, and then they get lost. If you do the math, there is a bit left, right? This would mean that there is net growth here. But there is no net growth. This ocean is not getting greener every time. So with the viral lysis, we're able to kind of like make the equation kind of fit. And if you translate that, it really says here, in a way, the nutrient-limited upper ocean in the summer, where you have these stratified, very nutrient-limited conditions. And in, for example, the tropic and subtropical, it's kind of whole year round limited. But in our regions, only in summer. What you see here is that whatever is produced per day is also lost per day. And that's quite essential in general. A lot of the students, a lot of people, we think it's not there. There's a barren ocean, I don't see anything. And if it's not there, then it's probably not happening. But it is happening. It's happening at a very fast rate, but it's also lost at a very fast rate. So there's a tremendous turnover the whole day going on. Okay, we know this. I'm a little like a, like a fast train going through. Tell me if I go too fast, please. I lose myself sometimes. What about the future scenario, right? I mean, we did this whole transect to see if there was actually an effect. Well, we plot here the ratio of viral lysis to grazing. So if this is the one-to-one line, then viral lysis is equal to grazing. If it is above the one-to-one line, it's a viral lysis dominated system. Doesn't mean that there is no grazing, it's just more viral lysis. If it's below the one-to-one line in the north, it's a grazing dominated system. So if we now extrapolate our findings from the south to the north in the future, we see that we actually go to a lysis dominated system, which I hope you kind of got that means we're going to a more regenerative system. There's going less to the higher trophic levels and more to this microbial food web. And that could mean, and I made the mistake once to say that it's less fish, period, no question mark. And that was at the Ministry of Economics. I kind of did that a little bit on purpose to tease, but I also realized, yeah, it's not good. You should actually, we don't know for sure if that's going to be less fish. But it kind of like hints towards it, right? I mean, I hope that you agree with me that these type of data really urge for including viruses in also the ecosystem models, in our global models, in trying to understand where is the energy going? And are we really as rich in the fish as we think? That's besides the overfishing. So let's go to the other example. Northwest. We could have done northeast, but that was just because it was part of a larger project. So we went northwest. And the question is, we're going in spring now, and the question is viral lysis as important as it was in summer? And this is a region, especially we're going a bit higher up here, this is a region like our temperate regions, right? The plants start growing again in the spring. The grass starts growing, we start to see leaves. This is the same in our coastal seas. We start to have higher amounts of phytoplankton. Blooms, we call them. So if that's there, and we just know the formula, net is gross minus losses, they either grow faster or the losses are less. And if the losses are less, is it grazing or is it viral lysis? Well, the fact that I actually tell you that means that they don't grow faster. Otherwise, we wouldn't go the web path, right? This is just a summary of all. We grouped them in different groups. That's for this talk not of interest. But what is striking in this graph right away is that the reddish bars are definitely smaller than the grazing bars. And I can tell you that the grazing bars are comparable to the summer rates. So it really means that we have less viral action in the spring. And if you now look at every point independently, you plot gross growth rate, so what they can, their production versus the losses. And again, this is a one-to-one line, and everything above the one-to-one line means that there is more production, more gross growth than there are losses. Which automatically means that we have accumulation. So it kind of shows that with our data, we are able to kind of like, show what is going on in the real world, but we also have been able to understand why, who is actually why. There's not a higher gross growth rate, there is less viral lysis. And we think there is less viral lysis because in spring the water is not so stratified yet, it's not so calm, there is not warm water layer on it yet. It hasn't been, we're still in kind of like the storms. So deeper mixing. And if you have 1,000 viruses over this area or over that, well, let's put it not an area, over this volume or over that volume, it makes a difference, right? The chance to bump into a host is less when you are deeper mixed. So this actually really explains, and the grazers can actually swim, right? They can search for their prey, the viruses can't. So it makes sense that in spring, this is the case. of course, when we have more climate change and we're going quicker to the warmer waters, we will also see that there's a shift towards the viral lysis again. These are two really cool examples, I think, of reduction of a bloom. This is the one we had in spring, right? Temporarily, they can escape viral lysis because, for example, they have a low virus number. This is supposed to be the mid part of the talk. Thank you so much. Cheers. Alcohol-free. I don't need alcohol. And it is good, by the way. Thanks. Oh, no, that's not smart. So low virus number, right? So reduced chance to meet each other. But it could also be, there is Phaeocystis again, that you make a different cell type. They can form colonies, which is a mucus matrix, a sphere with 10,000 cells in them. And each cell has a very highly reduced chance to be infected based on that sphere. They only see part of the cell, there's mucus, and the fact that this is a large entity versus a small really does the math. However, they cannot keep up staying in a colony because you need a lot of light for that and a lot of nutrients. So at some point, these cells are released and then these viruses hit. And then they get, this is the other example, this is more like example 1, right? This is the potential, but by severe viral pressure, you actually have a lot of growth, also a lot of losses, and then this is the resultant standing stock. So there is a lot going on. We just don't see it really. So the other point then here with this type of viral control, and this is in a way, this is a different concept, right? This is really an ecological concept. This is not the epidemic like with the COVID. That's really more, although, I mean, you could easily make that an ecology session, maybe for the Q&A session. But viral control and the virus production is dependent on the host. And the host is kind of like is regulated, dependent on the different conditions it's at. Different temperature, different light, different nutrients, as we already talked about. And if I just click in here, we talked about the morphotype, right? The colonies can strongly reduce the contact rate, just the fact that they actually are being infected. But we can also have what we said, the nutrients. We show that for especially nitrogen and phosphorus, limitation strongly reduces the production of viruses because these viruses have DNA and that contains a lot of phosphorus. And the nitrogen is needed because the proteins have to make these particles. So it makes sense that that is a reduced production. And there's a small catch there that's really interesting. These small-sized open ocean phytoplankton, they are very good in these limiting conditions. They can still grow fast. So they found themselves a niche, a place where they can grow fast and have reduced viral production. Right? They are able to kind of like outgrow their viral pressure because they don't make that many and they can still grow fast. You put a big cell there, he's not able to do that. So again, it's in a kind of like smart, if you want to say, to be small. We also look at light. We have an optimal light condition. We can put lower light, and that's since it's a photosynthetic organism, it gets energy limited, so it takes long, and they don't make that many. But also at high light, we see a negative effect, because then the cells get stressed. They make certain compounds that are not very good for the cell, and also therefore the whole production of viruses is affected. If you now, because this is actually, if you combine these things, can look at this, look at a dual effect, and we did that together with phosphorus limitation. It looked as if phosphorus limitation determined everything. Light was not really interesting anymore. But then we checked these newly produced viruses. We checked whether they were still infectious. And especially on their low light, we only have 5% of them still being infectious. So they produce all these virus particles, but they didn't really do a good job. And that's a little bit like, for example, with the flu, right? I mean, that's a single strand of RNA that is prone to making lots of mutations. There is no checkup system on it, and you just poop them all out. And some of them may actually do a better job. Most of them don't. If you have, in this case, double-stranded DNA, it's relatively conservative. There's a checkup on this thing. So that they don't make these infectious progeny to 95% loss, that is quite something. And it's important in a way for any discipline you're studying, if you're a student, but otherwise as well, not to think just on one factor and then say, okay, that's it. Then another factor, that's it. Put the factors together and you get sometimes really interesting but also weird results. We also put temperature and light together. And we did that for an Arctic system, small algae. And we had 4 viruses for that system, but we used 1. This is just to show this is ice cover. So this is just a season over an Arctic season, and temperature goes from -2 to 7 degrees. Seawater can go below 0, right, because it has so salt. And then we have highlight, for example, 24-hour light here in this period with also a relatively high temperature. And here, of course, under the ice, very low light and very low temperature. And we've been able, based on our results, to try and understand, predict in a way what's going to happen over such a whole season in the different layers of the water column. And I was really happy. But then you realize there's also something missing. Because viruses don't stay in the water forever. You know, some of them get UV dosed, they get sunburn, their DNA is getting damaged. Then you have some smart viruses that actually still infect and tell the host to repair it. But you know, the things happen. There is pollution. We've shown that oil really is not very good for infectivity. We have here shown that dilution by mixing, right? I mean, that's not on infectivity or loss, but it's temporarily not being able to infect. And here, aggregation, these bloom-forming algae with all their dead cells, and the water moves a little bit, makes little aggregates, make little clumps of material. Looks a little bit like, you know, when you mix an egg, but that becomes foam, but you get these kind of like strands of organic material. And if you have that, they can be very sticky, especially when it's under a phosphorus limitation. And then a lot of these viruses, up to 75% of these viruses, were just instantly attached, passively absorbed to these aggregates and not available. So this is kind of like a funny way almost, you know, there is no intelligence in these unicellular organisms, but it's almost like, you know, got you. I produce you, but you're not getting anywhere. This is another really cool example where we have a breadcrumb sponge and we gave it viruses, and within a quarter of an hour, it was gone. And we gave it again and gone and again and again. You know, as long as the PhD student could last. And it was her choice. It was just instantly, right? These sponges, this is actually not a breadcrumb sponge, doesn't look like that, but it's pumping water and it's filtering out, and we show that it's filtering out these viruses very, very efficiently. So there's potential application if you want to think about that. But the Netherlands doesn't really have a lot of coastal, rocky coast, because they need to be on something hard. But still, it could potentially be used. Very, very efficient. But it is hard to grow sponges, by the way. These particles we show here in the Arctic, these glaciers are melting. And if you have ever visited a glacier, there's quite some sediment on it. There's erosion, but there is also some, you know, like just sediment that is blown in. It's relatively reactive sediment. There's not a lot of organic material already on it. And it really takes out the viruses from the water column. It cleans more or less the water column very efficiently again. This is temporarily, but for the algae that are there, they can actually grow potentially a bit better. And here we looked at, and also the same Arctic viruses again. We wanted to know, like, okay, if this is going from 0 degrees to 7 degrees, do they actually have the same infectivity? Is temperature affecting how efficient they are? And to our surprise, we have 4 viruses isolated at randomly from more or less the same water. And we show that 1 is very good at 7 and very bad at 0. 0 is melting ice, right? So it's 0.2 probably. And we have also a virus that really was the opposite. So we've got this really interesting dynamics, and this is just one host. So imagine if you have different hosts. This is a real good example of how biodiversity is also within that system going on. Not just you kill a species and then another species gets a chance. No, here really is, they have their own specialty in a way. But back to Antarctica, right? This is the third field work, and then we're almost done. Then we can go home and measure the 16,000 samples. This again is the same, and you know, the Arctic, the Antarctic, there is ice, there are some algae, and then we have phytoplankton and big grazers. Traditionally, people thought, most people, if you look at textbooks, if you look at polar ecosystems, polar oceans, it's a very short food chain. Big algae, and then you have big small critters, and then you have small fish, big fish, and then there's the penguin. It's a very short food chain, but that's not true. And actually has a lot of small algae there as well. And with the retreating ice, there is also a prediction that there will be a community change in these algae, and they will go towards more smaller sized algae. And that will have an effect on the length of the food chain. And since every step in that food chain is taking its own part of the energy and carbon, we're ending up with less fish because of that as well. And we have no clue if this is also really affecting. Is viral lysis another factor that is contributing to less fish in a way? So what we did is we went to a base, Rotorua base. It's from the UK. There are no claims on Antarctica, but their field stations are run by different countries. And then you have a rubber boat, and if the weather is kind of good, then this is where you speed up to your sampling point. They have a monitoring point, there's some coordinates, that they already do for a lot of years, so we are joining them. And we took it for the whole full season. So that's about, and we were really lucky, we could really stay long. This is to make you jealous at the end of such an evening, right? That was Ella for 5 months, enjoying the view. And you have to realize these are the photos when the weather is good. Yeah. This is the Dutch docking station, and it has 4 rolling doors in which you can put a lab container. And these lab containers can be very specific on what you need. We need very low temperature. My neighbor needed ultra clean conditions. Another one needed radioisotope. So this is really interesting. The mobile labs. It's really handy because you can exchange them. The other reason they're mobile is because we can't leave anything on Antarctica because then the Dutch claim a territory and we don't. So, but we were really lucky to have this association, this connection with the English people, with the UK, a Rotorua research base. So anyway, this is very nice weather, but this is not sampling weather because there's too much ice and the rubber boats can't go through it. And sometimes there is a logic building, you also had to cut your way out when it's in the early season. It's kind of funny. And you do really think cool things. I mean, I have a license for crane, I have a license for boating, I do ski-doos. I have an international license for boating because I sample in Spitsbergen, Svalbard. And you use it all once, right? You go here, I have a bit more EHBO. Don't know what it is in English. First aid, higher level. And you use it all very, very rarely. So after that, you're not allowed to use it. So it's kind of like really cool. You need to learn all this. So let's go to data. That's what I do daily, and I can keep doing that. The same colors. Blue is grazing, red is lysis. These are just 6 of these groups, but now this is time. We're going from December, this austral summer, we're going from December to March, right? So in November we arrived, dig out in the snow, and in March the last plane was leaving before the overwintering. What I would like to show you here is these extra gross growth rate circles. So for example, here you can see all the circles are above the bars, right? Which means we have more gross growth rate than losses, which would mean we have accumulation, remember? And we see that in the net standing stock. This is nice. You always want to try and have independent methods being comparable. Here we have the opposite. The circles are lower than the total loss, and we actually see a decline in the standing stock. Here we have the situation as if we were in the summer. We have approximately, sometimes higher, sometimes lower, but approximately the growth, which is really high, it's more than a doubling a day, is matched by the losses. And look, the standing stock is really low. So for the different groups over the season, different times of the year, sometimes it's a bit more lysis, sometimes it's a bit more grazing. There's all these different things we can kind of like tweak out. But overall, we have a very dynamic season, and it's all related to these environmental factors, to the temperature, to the salinity, to the light and the nutrients, even in Antarctica. And yes, low temperature doesn't stop viruses, because these hosts are actually optimized for low temperature, right? So these viruses are optimized for their host. So it doesn't stop because we just go to low temperatures. This is the last science part. We have to convert everything to cellular carbon, because that's, in a way, the currency. If you want to do an ecosystem model, if you want to do a food chain, that is what this, because these are rates. But we don't know whether there is a lot of them. We don't know how big they are. And it could be that we have, for example, a few big ones, and if they lyse, their cell content is definitely going to, be a large portion of the carbon that we need compared to smaller ones. So we need to translate it to carbon. We take into account the abundances, how many, and we take into account their cell size. And what we show then is that viral lysis is critically important to actually get a closing seasonal mass balance. At the end of a full season, you're not left with something. We don't have, well, there are some plants that stay green in the winter, but they don't really grow, right? We don't build up every year more biomass. At the end of a productive season, it kind of also, what is grown is also lost again, one way or the other. And this is what we also show here. So in contrast to general belief, we really need these viruses also in the Antarctic. And for you, this might be all kind of like, oh, okay, she tells me that. But this is really, for us, it's like, well, finally we have proof, right? And it also means that it directs a lot of the carbon away from the penguins. You have to realize that is the current situation. We just didn't realize that, right? We're not all of a sudden having less penguins, right? This is just that we now finally understand how the penguins at the moment are run. There could be more if we had less viruses, but maybe that would give other problems. For example, also our gut, right? Everybody knows now that our gut is full of bacteria. If there would be no viruses, it would be very full in our gut, right? So viruses also have good purposes. They really do help to control. And by the way, the fact that we're mammals is due to a viral gene. Yeah, so we got a piece of viral genetic code that allows us to allow the fertilized egg cell to actually, adhere to a wall and make a placenta. So that whole idea is viral. We have about, I thought, out the tip of my head now, because I thought it was 8% or so viral DNA in our genome, and it can really be good. It can also be devastating if you're the cell or the human that is dying, of course. But ecosystem-wise, ecology-wise, it's all good. So I hope that I convince you that the collective impact of viruses in the world, but definitely in the oceans, is huge, right? A, we have a lot of water, but B, we have a lot of viruses and they do a lot of things. We have at least 10 different viruses per species. So they seem to be very simple, but actually their power goes way further than their just initial destructive power. They are absolutely essential for biodiversity on the genetic level, on the species level, on the intraspecies level. They are essential for driving these biogeochemical cyclings, these elements, these carbon, nitrogen, phosphorus cycles. They actually keep control on bacteria, also the bad ones. So they're very important. If you buy a house, people always say it's always about location, 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 right? I mean, and that's true in my research, by the way. I'm the lucky one, right? I have done Antarctica, or that way, and I have done the Arctic. And so, yes, I am very lucky with that. But I think what is really important is that it is about perception, 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 right? We should always dare ourselves to look from another angle. And because the first one who saw viruses did not really, I mean, I have a double-stranded RNA virus isolated. I wanted to have the double-stranded DNA virus. The damn thing was still in that sample. And at some point, it was like, yeah, but somebody else already has a double-stranded DNA. Maybe I should check out this one. Right? And it turns out that is awesome. And we thought, actually, that at the time, it was not sequenced. We don't know the full genome. And we thought, like, oh, you know, this belongs, the closest relative is a lethal diarrhea. It causes lethal diarrhea in kids. You could see, you know, you could see already my boss was like, okay, flags up, you need to secure everything, you need to kind of contain it. And then we sequenced it, and of course it was something very different. But it's at the origin, again, of a split of these viruses. So it is super cool to kind of like think outside the box or grab your chances when you can. And this was just fun, because I stood in the rubber boat, as you can see here, in Spitsbergen, and I just did this, and my colleague took a photo, and she says, now me. And we have spent 15 minutes to get it, right? And this was chance. It was the right photo. I definitely want to thank everybody in the lab, because, I mean, seriously, PhDs and postdocs do most of the work. I try to guide them, but they are the brains. I thank the technicians and, of course, all the ship's crew and the base crew. That's essential. Without them, there is no work. The collaborators, the people that we work with, are just, it's awesome always. And, of course, the funding agency. And in my case, that is mainly the Dutch Research Council, NWO. And with that, I thank you for your attention.